And before that, I would like to briefly introduce Dr. Kudo by using this slide. Okay. Dr. Kudo was born in Yamagata Prefecture, Japan in 1951. He received his bachelor and master degrees from Tokyo Tech and doctoral degrees from the University of Tokyo in 1985. He has published more than 160 journal papers, and he is now Institute Professor and Professor Emeritus of Tokyo Tech and visiting professor of Showa University. His research specialty is bone biology. Okay, Professor Kudo, please come to the stage.
Thank you for your kind introduction. I'm Akira Kudo, a professor in Tokyo Tech. Today, I'm going to talk about Medaka fish in space. Actually, all animals, including Medaka fish, have been adapted in the environment of Earth. That is 1G gravity. But today, Medaka fish are going to another world. It's microgravity. Okay, let's start my talk. Today's my seminar title is Medaka fish went to space, bone loss in the International Space Station. So this scene is a launch of Soyuz rocket in the night. So we booked the Medaka fish here. Okay, so bone, we have a problem of bone under the microgravity. We have a bone weakness in astronauts. And under the microgravity, we have a major two problems in our health. One is bone weakness. The another is muscle atrophy. And about bone weakness, so before going to International Space Station pre-flight, bone mineral density is 1.01 gram per cubic centimeter. But when you went to the International Space Station, this bone mineral density was decreased to 0.95 gram per cubic centimeter. So in such a case, we can speculate the osteoclast is activated because the osteoclast is bone resolving cells. So I raised one question about our project. How are osteoclasts activated in space? So before we are going to my experiment, I would like to explain the background of this experiment. So bone loss under the microgravity is strongly related with disease of osteoporosis, specifically age-related osteoporosis, osteoporosis for old men and women. This is a normal bone, backbone. Usually, all human bones are replaced by all human bones are replaced by new bones within 5 to 10 years. But if you have a problem of this control, so you have a disease of osteoporosis. So right side shows on the backbone of a patient. As you can see, you can see a lot of holes inside of backbone. Such a hole can make a bone fracture. So it's a very severe disease. And osteoporosis in the world, 1/3 of women and 1/5 of men become patient. It's a very severe disease. To see what happens on the on bone under microgravity, we perform 2 experiments for reviewing the mechanism of bone loss in ISS, International Space Station. One is the project name is Medaka Osteoclast. 2 months rearing of Medaka fish launched by Soyuz TMA 06M in October 23, 2012. Second one is Medaka Osteoclast 2, 1 week observation of fluorescent signals of osteoblast and osteoclast in Medaka lab in February 2014. This osteoblast is bone forming cell. So to perform this experiment, so we need an International Space Station. This ISS provides the environment of microgravity. Just I say microgravity. So usually in ISS, so the gravity is 10 to minus 6, 10 to minus 6 G. We call it microgravity, but it's nearly zero gravity. And also which fish apparatus, this was installed in the Space Research Laboratory KUDO. And also we utilize Medaka fish. The scientific name is Orygius latifis, is the first vertebrate for an animal model in ISS. Here, so I would give you one question. So one question, please. First question, please. So we have an International Space Station. My question is one, how far does the International Space Station fly over the Earth? Answer A, 400 kilometers. Answer B, 800 kilometers. Answer C, 1,600 kilometers. I will give you 1 minute. Please choose one answer. Okay, please start to answer. Okay, maybe this is very easy answer. Okay, 1 minute finished. So okay, let's see your answer. Okay, answer A, we have a 38%. Answer B, 35%. I think all, so all answers are even. So correct answer is A. So surprisingly, so just only 400 kilometers over. So it means that in the night, by your eyes, you can look this International Space Station. Okay, let's go next slide. So just I explain about the bone structure of Medaka. So we have here in this side, we have pharyngeal bones and teeth. Actually, we have more than 500 teeth in this area, very small teeth. And for supporting these teeth, we need pharyngeal bones. So just I explained, the osteoclasts are localized on the pharyngeal bones. So just I introduced what osteoclast is, the property of osteoclast. So this is osteoclast, and the typical character is so bone resolving cell. And also we have osteoclast have many nuclei in one cell. Normally, so you have only one nucleus in one cell, but as a typical feature of osteoclast, you have more than 10 nuclei in one cell. For example, for the Medaka fish. Also, so you please remember that this TRAP, this is the osteoclast marker, tartrate resistant acid phosphorus. This is enzyme. So this is a good marker for osteoclast, and this big osteoclast sit on the bone, and also this osteoclast can induce the acidic condition here, and to resolve bone. Just you remember so TRAP. So to see where we have osteoclast in Medaka fish, we perform the TRAP staining, enzymatic staining. this red color shows where where osteoclasts are. So as you see in this pharyngeal region, you can have many red colors showing the osteoclast. So therefore, we focus to see osteoclasts in this region under the microgravity. To visualize the osteoblast and osteoclast, we established the transgenic line. It's a gene manipulation. And for detecting the osteoblast, so we utilize Osterix gene promoter combined with DsRed protein genes. So you can see osteoblast in the red fluorescence. And in the case of the osteoclast, you utilize TRAP again, and osteoclast marker linked with GFP protein gene, and you can see osteoclast in green color. So finally, we established a double transgenic line and to see where we have osteoclast in Medaka fish. So here I would like to introduce a living Medaka fish. So now we have 3 Medaka fish here, and we have 1 male and 2 females. Can you distinguish male and female? So if you would like to distinguish, you should use analphine. So bigger analphine means male, and smaller analphine means females. And so such Medaka fish, they live in East Asian countries. And so I will sample 1 Medaka fish and put on the slide glass. And this is a fluorescent microscope. I put this sample under the microscope. So you see this beam. So this beam, this laser beam can activate protein or fluorescent protein to see fluorescence in red or green color. So usually, actually this machine is a little bit expensive, but in Tokyo Tech, faculty student can use this machine for the experiment. So let's see the fluorescent signals by using our photo. Okay, so this is whole body of Medaka fish. So you can see the red color and the green color. So red color means osteoblast, and green color means osteoclast. So this is a backbone, but we should focus to see the pharyngeal region. So we go to the pharyngeal region. So here, there are a lot of teeth in the pharyngeal region. Also, you can see the many osteoclasts here in the green color, and osteoblasts in the red color. So let's go back to my slide. Okay, so we are going to the International Space Station, so we need a crew training. So he's astronaut Hoshide. He handled fish carrier. So this fish carrier comes from Baikonur, and so he would like to put some Medaka fish into the tank. So this scene was recorded around 7 months before launch of the Soyuz rocket. And experimental protocol, I would like to introduce my protocol with timetable. And we collected 3,000 eggs in Japan, and took 300 juvenile Medaka fish we took to Baikonur. And in Baikonur, we are breeding around 2 weeks to select only good Medaka fish. And after going to space, 2 days in space, we sampled 8 fish. And 2 weeks in space, we sampled 6 fish whole osteoclast. This 8 fish, we utilize this 8 fish whole RNA extraction. And also 2 months, finally 2 months in space, we sampled 6 fish whole osteoclast and whole fish whole RNA extraction. Put together, so those osteoclast samples, they came back by Soyuz again. And these RNA samples, they came back by SpaceX, Dragon SpaceX. So these are apparatus for our experiment, fish carrier and fish catcher. You can sample fish by using this one, and the fish fixation. And in the International Space Station, we fixed all fish in ISS. And also, we have 2 tanks of Medaka fish in multi-purpose small payload rack. So we have 2 tanks. So the size of 1 tank is 700 milliliter. So you can have 8 fish for 1 tank. And we have 2 tanks. It means that we have 16 fish, Medaka fish, in the International Space Station. And here is Baikonur. This is a launch site of the Soyuz rocket. This Baikonur located at the center of Asia, and it takes 4 hours by airplane from Moscow to Baikonur. And this is a breeding room. So during 2 weeks, we must choose only good Medaka fish. So here, so I have a next question to you. By using this apparatus, we must choose only good Medaka fish. So could I have a second question? So we must test an important ability for Medaka fish to let them adapt to microgravity, since some Medaka fish twist or turn around in space. So second question is, Medaka fish are twisting and turning around under microgravity. To prevent this abnormal behavior, we examine the physical ability. Which ability did we examine? So answer A, ability of eyesight. Answer B, hearing ability. Answer C, ability of motion. I can give you one minute again. Please give me your answer. Okay, start, please. Start to answer, please. Okay, I can wait for 1 minute. So this question is not so easy, but please consider to block the twisting or circling under the microgravity, which ability is important. Okay, let's see your answer. Okay, very interesting. So the majority is C, ability of motion, 76%. Maybe, I think it makes sense. But the correct answer is A, unfortunately, ability of eyesight. In the selection of good fish in space, the most important ability of fish is good eyesight. To test eyesight, we prepared a small apparatus here, having turning white and black color paper. Fish look this black or white color. So I show you this testing movie. So we put 1 Medaka fish at the center of apparatus, and we turn this black and white paper by increasing speed. And the Medaka fish can watch this white and black and white color, and the Medaka fish will circle here. Okay, let's see the movie. So now you have a Medaka. So the Apparatus. So now so when circle is enlarged, so Medaka fish cannot catch up the white and black pepper. So this Medaka fish is totally upset, finally. And the speed goes down, so Medaka fish can stop or not. Okay, it's good. So Medaka fish can stop. Sometimes, so some Medaka fish cannot stop. So, but unfortunately, we cannot take this Medaka fish into ISS. Now, this Medaka fish is in the Baikonur. It's okay. Next. So one day before departure, so we looked Soyuz rocket. Here is a tip of Soyuz rocket. So we booked 24 Medaka fish with 3 astronauts. So it means it's very crowded. And on the departure day, so we have a ceremony for 3 astronauts. So he is Mr. Ford. From NASA, he supported our experiment. So this is a launch of Soyuz rocket. So just we would like to see the movie of this launching. Okay, let's start. You see the Soyuz rocket here. Arm is off. Higher. And large. So that day, so we had a very clear sky, and it was announced that under 5 minutes after launch, so this launch was successfully performed. The Medaka fish went to space. So now, so we have a movie here. So this movie is 10 days after staying International Space Station. So this is, this tank. So we have a feeding apparatus here. So it's surprisingly, so we have big bubble here. So this is a sliding door. When this door opens, so Medaka fish can eat food. But unfortunately, Medaka fish cannot eat because under this big bubble blocks. So I give you last question to you. So we have a question of how Medaka fish could eat food. Please put your answer in chat in Zoom webinar only. So please send your chat answer to everybody, to me or also other people. So I can give you 2 minutes for getting answer. So please put your answer in chat. We have a 300 chat here. Still, I'm waiting to get your answer. Slow mass. Slow mass. Of course, yeah, you can, Medaka can eat food by mouth. So this is true. But now you have big bubble. So Medaka cannot eat food. By putting the food, okay, slow skin. Slow skin. So it's very difficult to, pass the food to the inside of the body. So it's very difficult. Slow nose. So my question is, you have a big bubble at the entrance of the door. So when door opens, you can see food. But under this big bubble blocks this entrance of there, this entrance of door. So you need to take out bubble. Absorb through skin. Eats anything. Sorry, through the part of photosynthesis. I don't know photosynthesis. So the through gills. Yeah, ear is okay. So we 2 minutes now is over. Now it's over 2 minutes. Okay, thank you very much your answers. So let's go on. Okay. Let's go on. So from the movie, you can see the correct answer. Okay, let's start this movie. So now you see very strange, moving here, circling something. Now the sliding door opens. But so Medaka cannot eat food because the food is here. But you can see the many small bubbles here. But this big bubble becomes smaller. It means the Medaka fish can drink a portion of the bubble. They spit out into the water. So that reason why we have many small bubbles. But this bubble, the size is decreased. So finally, all Medaka fish can eat food. So it is very surprising to me. It is very surprising to me. It means that all 8 fish can together can work together to make smaller bubble. So they are very good Korean. So that time, so I can say that maybe this experiment is successfully finished. Okay. So you see the correct answer. So Medaka fish work together to make this big valve smaller. Okay. And second movie, at day 47 in space, fish became motionless. It's also very interesting behavior. So we start a movie, but before that, so we have 4 Medaka fish here. Please focus to see this fish. So this is movie, not photograph. Okay, these two fish can move. Also, this fish start to move. But you see, this fish can't move. This is water. It's very strange. I think that, so after 7 days under the microgravity, Medaka fish can be adapted to the microgravity because they don't want to move. It's not necessary to move. Okay. So for mineral density, for the mineral density, what happens? So just I will show you the data of decrease of mineral density. In the soft X-ray analysis, so if you compare to the fluoride sample with ground sample, so you focus to the pharyngeal region, so this mineral density went down. And also in the micro CT analysis, compared to between the ground sample, also the fluoride samples, if you find the red color, it means the mineral density is high. If you see the blue color, it means the mineral density is low. Compared to the ground sample in fluoride sample, so the red color is gone, and you see the green and the blue colors. It means the mineral density is decreased. So we should conclude that 24% of mineral density is decreased in 2 months flight. So now, you see the fluorescent signals of osteoclasts. This green color shows fluorescent signals of osteoclasts in the fluoride sample. So these signals become stronger. So you see the osteoclast activity is enhanced under the microgravity. So also, we performed RNA extraction. So we carried back the samples by Dragon SpaceX company, and we extract the RNA, and also we performed the DNA sequencing. And by using this technique, so we detect very interesting 2 genes which are highly elevated transcription under the microgravity. One is FKBP5, the another DTIT4. This is a summary of the first experiment. We could find enhancement of osteoclast stability in space. So this is osteoclast in space. So the number of nuclei is enhanced, and also the volume of osteoclast is enhanced. After that, we have a high ability, high osteoclast ability. They can resolve preferential bone. So we quickly go to the second experiment. So first purpose of this experiment, we would like to see the fluorescent signals in the International Space Station. So to see those signals in ISS, we prepared 6 different transgenic lines to see early osteoblast, osteoclast, and stress-induced indicator. So in the combination of those transgenic lines, we booked 12 Medaka fish in a sample chamber. This is sample chamber. So you see 12 different transgenic lines. And by using the fluorescent microscope in ISS, so we put this sample chamber under the microscope, and by remote control, we could observe the fluorescent signals in Japan. This is the final result. So fluorescent signals of osteoclasts and osteoblasts in the pharyngeal region 5 days under microgravity in ISS. So those signals are quite enhanced. So this red color enhanced, also blue, green color so enhanced. Okay, this is final slide. So thank you very much for your joining us, and I hope that so next time is you. So please go to the space station or space in the close future. So thank you for your attention.